Hi everyone, I'm John. Today I'll provide a high-level overview of my thesis entitled Enabling Fast, Accurate and Efficient Real-Time Genome Analysis via New Algorithms and Techniques. Let's start by looking at some key applications in genome analysis. For example, we can uncover and treat diseases linked to genomic variations, alter genomes to solve fundamental challenges of life, detect pathogens in the environment, and by doing so enable rapid surveillance of disease outbreaks. To enable many of these applications, we need to analyze the genomic data. The analysis of genomic data starts by converting the biological molecules into digital nucleotide sequences that we call reads. However, there are certain challenges to analyzing genomic data. First, the genomic origins of these reads are lost after sequencing. Second, we need to analyze a large volume of data. Generating the genomic data is usually the first step in a common genome analysis pipeline. And initially, these sequencing devices produce raw sequencing data, which either needs to be translated into nucleotide sequences with a process called base calling, or pre-processed to reduce the inherent noise in raw sequencing data before further analysis. To identify the origin of sequencing data, we compare it with a representative sequence of a species called the reference genome. This process is known as read mapping and involves several steps. First, we build an index, a hash table from the subsequences of a reference genome as a target sequence. Second, we extract subsequences called seeds from the sequencing data and query the index to find seed matches in the seeding step. Third, we identify nearby seed matches called chains to locate potential genomic regions of origin. Fourth, we optionally find exact matches and differences within the chaining region. The information we generate from read mapping can be used for several purposes in downstream analysis, such as genome assembly, variant calling, and metagenomics. We find that the imperfections both in sequencing data and the analysis of this sequencing data negatively impact the entire genome analysis pipeline. For example, we have unwanted variations in raw sequencing data and variations that are left unhandled when analyzing base code sequencing data, which we refer to as noise. Noise causes significant computational overhead, limiting the accuracy and application scope of genome analysis. To address this problem, my thesis statements as follows. We can mitigate noise in sequencing data and analysis by building a better understanding of the types of noise and developing new algorithms and techniques that can tolerate and reduce noise, thereby providing accurate, scalable, and real-time analysis of sequencing data and enabling new applications in genome analysis. Now I'll present the core contributions of this thesis in terms of the types of noise they address and the new applications they enable. Our first contribution, blend, addresses noise in the analysis of base code sequencing data by providing an effective hash-based search method for noise tolerance. Second, RawHash targets noise in raw nanopore sequencing data to reduce noise and enable hash-based search for raw signals. Third, RawHash 2 improves our understanding of noise in raw sequencing signals, allowing for better noise reduction. Our fourth contribution, Rawsamble, enables a new application by overlapping raw nanopore signals to build assemblies without base calling. I'll start by discussing blend. Let's start by looking at the traditional mechanisms we use for finding seed matches between a reference genome and a read sequence. First, we identify certain subsequences or segments and generate their hash values to use as seeds. Second, we find seed matches if their corresponding sequence segments exactly match. This hash-based mechanism enables fast and memory-efficient exact seed matching, while ensuring that hash values from dissimilar sequences are unlikely to match, avoiding false matches. However, large segments or seeds are unlikely to exactly match due to potential mutations and sequencing errors, which we call noise. This leads to generating completely different hash values due to the nature of low collision hash functions used in seeding, which prevents us from identifying highly similar sequence segments with quick matching. The common solution to identify such highly similar regions is to reduce the seed length used for matching, which can help avoid non-matching regions. However, using shorter seeds is problematic as it results in a larger number of shorter exact matches to analyze, leading to computational overhead and inaccuracy. Our goal is to enable generating the same hash value for highly similar seeds while generating different hash values for dissimilar ones, which we call fuzzy seed matching. To do so, we replace the existing low collision hash functions in seed matching with a hashing technique called simhash, which can generate the same hash value for highly similar vectors and different hash values for dissimilar ones. However, due to its mechanism, we cannot directly use SIMHash to generate hash values from seeds. To enable the use of SIMHash and its properties, Blend provides effective sequence-to-vector conversion strategies. Let's look at our evaluation methodology. We integrate Blend into Minimap2 to perform end-to-end read mapping. We use real and simulated datasets from various sequencing devices, including highly accurate reads from PacBio Hi-Fi. We evaluate Blend in terms of two use cases, read overlapping and read mapping. Before discussing the results, we first conduct an empirical analysis of Blend's fuzzy seed matching capabilities. We calculate the cumulative proportion of hash collisions on the y-axis based on the edit distance between the seeds with collisions on the x-axis. The goal of Blend's fuzzy seed matching is to increase this proportion for highly similar seeds at lower edit distances. Here we show the proportion of hash collisions at certain edit distances for Minimap2. We find that only a small portion of seed pairs with hash collisions are similar. We observe that Blend increases the proportion of hash collisions between highly similar seeds, and Blend can be configured to further improve this collision rate. We find that Blend enables fuzzy seed matching by systematically increasing the collision rate for highly similar seeds. These figures show CPU time, peak memory, and assembly accuracy when using highly accurate reads for overlapping. Compared to Minimap2, Blend provides a speedup of up to 84 times, reduces peak memory requirements by 14 times, while providing the most accurate assemblies generated from the overlaps identified by Blend. We conclude that effectively tolerating noise improves both performance and accuracy. This concludes our first contribution, Blend. Motivated by the potential to improve both performance and accuracy by tolerating noise effectively, we switch our focus to an analysis where we have significantly increased noise. For this, we look at raw nanopore signal analysis in our second contribution, RawHash. Let's look at how nanopore sequencing works. This is a nanopore sequencer that contains many tiny pores called nanopores. As a nucleic acid molecule passes through a single nanopore from the negatively charged side to the positively charged side, we can measure the ionic current disruptions that are distinctive to identify each nucleotide passing through. These disruptions are generated as raw electrical signals at a certain throughput. We can analyze these raw signals instantly as they are generated, which we call real-time analysis. And based on the real-time analysis, we can stop sequencing a read or the entire sequencing run. There are two main benefits of real-time analysis. First, we can reduce latency by overlapping analysis with sequencing. Second, we can reduce sequencing time and cost by stopping the sequencing early. There are two main ways of analyzing raw nanopore signals in real time. The traditional way is to translate these electrical signals into nucleotide sequences with a base calling step that mainly utilizes complex deep neural models. These base called sequences can then be analyzed using regular read mapping steps. An alternative way is to avoid the chorus-to-base calling step by direct analyzing raw signals. This approach typically starts by converting the reference genome into its expected electrical signal values using a pre-constructed conversion model followed by the analysis of raw signals. However, raw signal analysis is noisy. Let's look at why. Ionic current disruptions are caused by the presence of a certain small number of nucleotides inside a nanopore called a chimer. It is possible to sequence this short chimer at different times or in different nanopores. Each will report ionic current disruptions as an electrical signal measurement. However, noise causes slight differences in raw signals from the same chimer. This means we cannot directly match raw signals as this is not feasible. And even if we could, a single chimer is too short and likely to appear in many places in a reference genome. To enable assigning the same values to similar raw signals, the key idea in RawHash is to perform quantization in two steps. First, the quantization step creates a certain number of equal width buckets across the entire raw signal range. Second, we assign similar raw signals within the same signal range to the same bucket. This reduces noise by quantizing raw signals into equal width buckets. By doing so, RawHash enables matching raw signals by eliminating slight differences. Although we're limited by the length of chimers we can sequence at a time, we can still increase the chimer length used for hash-based search. To do this, we use multiple raw signals generated when sequencing a longer sequence segments from consecutive overlapping chimers. These quantized values, shown in binary, can be packed together using a simple bitwise operations and hashed. Our packing strategy captures both the content and order of multiple consecutive chimers, providing more detailed information than a single quantized value. We can reliably use the hash value after packing to find fewer and more accurate matches in the genome. by following regular steps in hash-based read mapping, including seeding, chaining, and mapping. Effective noise reduction enables us to perform accurate and real-time analysis in several steps. First, we store the hash values we construct from the reference genome and use them as an index. This is an offline and one-time task. Second, in real time, we query the hash values we generate from streaming raw signal data, which identifies matching regions between the reference genome and a read based on hash matches. Third, we use a classical chaining and mapping process to determine if a read is mapped based on the chaining information we have so far. If we need to sequence more to answer this question, we retrieve the next chunk of streaming data and perform similar computation in real time. If not, we can stop sequencing, reducing sequencing time and cost. We quickly show our initial comparisons of RawHash with the state-of-the-art tools Uncode and Sigma. We find that RawHash achieves a throughput speed of around 26 times and 3.4 times compared to these tools, and up to 1.75 times better accuracy when mapping reads to large human genomes. We find that effective noise reduction improves both performance and accuracy. This concludes our second contribution. Now I'll discuss our third contribution, RawHash 2. In RawHash 2, we enable a better understanding and handling of noise in raw signals, which allows us to further improve raw signal analysis. Although constructing equal width buckets enables noise reduction in RawHash, we find that equal width buckets leads to unbalanced loading, which can cause more false hash matches due to reduced uniqueness, impacting several parameters related to accuracy and performance. The key idea in RawHash 2 is to generate buckets with varying sizes, with the goal of balancing the amount of raw signals placed in each bucket, which we call adaptive quantization. By doing so, we can reduce collisions caused by skewed raw signal distributions. RawHash 2 provides three other key improvements over RawHash. First, rather than using a simple chaining score threshold, we generate multiple features to collectively decide if mapping should continue. We assign weights to these features and update the weights to optimize mapping accuracy and performance. We believe this idea should not be limited to raw signal mapping, but should be utilized in general read mappers. Second, we use sampling strategies to reduce the amount of raw signal data we process with frequency filtering and minimizer sketching. For example, the frequency filter excludes matching regions if the number of matches from a hash value is above a certain threshold. Otherwise, these matching regions proceed to steps such as chaining. Third, we improve the chaining algorithm with more sensitive scoring functions. We modify the earlier RawHash pipeline by integrating adaptive quantization, followed by the same hashing technique from RawHash and the new sketching step. We use the hash values after sketching to construct the index and query the index as before. Matching regions we receive after querying are filtered with a frequency filter, followed by a more sensitive chaining mechanism, and we decide on the mapping based on a weighted mapping strategy. We evaluate two different versions of RawHash 2. First, the default case without any sketching technique, and second, RawHash 2 with the minimizer sketching technique. We compare RawHash 2 with Uncode, SigMap, and RawHash, and we evaluate based on three different use cases, including read mapping. Let's first look at throughput comparisons. A single nanopore generates roughly 4,000 to 5,000 signals per second, corresponding to around 450 bases per second. A single nanopore device contains around 500 to 2,500 of these nanopores. We define computational throughput as the number of bases a tool can process per second per CPU thread. A tool's scalability is based on how many nanopores it can process with a single CPU thread. First, we show the throughput results for Uncode and SigMap, with different datasets on the x-axis and the throughput on the y-axis. The number of nanopores they can process with a single CPU thread is shown inside the bars. SigMap falls short of real-time analysis as its throughput is below the data generation speed of a single nanopore for human genomes. RawHash achieves significant speedup due to its fast hash-based search, while RawHash 2 and RawHash 2 Minimizer further improve throughput with effective sampling strategies. RawHash 2 provides an average speedup of 26 times, 19 times, and 4 times compared to Uncode, SigMap, and RawHash. RawHash 2 Minimizer further increases throughput by 2.5 times compared to RawHash 2. Next, we'll look at the mapping accuracy by identifying the correct mapping positions that each tool reports based on the ground truth mapping positions and calculating their F1 scores accordingly. This figure shows the mapping accuracy results on the y-axis using the datasets on the x-axis. The accuracies of Uncode and SigMap significantly drop for the human genome. RawHash provides comparable accuracy on smaller genomes and substantially better accuracy for the human genome, while RawHash 2 and RawHash 2 Minimizer substantially improve accuracy with the new and effective quantization and chaining mechanisms. We find that RawHash 2 provides the best accuracy in all datasets, and RawHash2 minimizer provides mapping accuracy comparable to RawHash2. By looking at both performance and accuracy results, we conclude that better understanding of noise can be utilized to further improve performance and accuracy. This concludes our third contribution, RawHash2. Let's now move on to Rawsamble, where we enable a new application, raw signal analysis, by utilizing the improvements provided in RawHash and RawHash2. Although reference-based raw signal analysis provides accurate mapping, reference genomes are not always available or ideal for certain applications. In such cases, we can construct an individual's genome from scratch by finding overlaps between reads. However, we cannot do this without base calling. Identifying overlaps between raw signals with existing hash-based seeding mechanism is challenging because it requires identifying hash matches when both signals are noisy, finding many useful overlapping pairs that we call all versus all overlapping, and generating long paths from useful overlaps. To address these challenges, Rawsamble provides several key improvements in raw signal analysis. First, to reduce noise when comparing noisy raw signals, we apply aggressive filtering. This removes similar signals that appear consecutively, often generated by sequencing errors such as stay errors, though it may result in some data loss. Second, overlapping reads can map to each other cyclically when using the target signals as query and vice versa due to shared regions. To prevent cyclic overlaps, we perform a simple deterministic comparison and report each overlapping pair only once. Third, we identify highly accurate chains to effectively prune incorrect overlapping pairs. Fourth, we enable constructing an index directly from raw nanopore signals instead of reference signals. We integrate the improvements made in Rawsamble into the RawHash2 pipeline as follows. We perform aggressive filtering on both target and query signals, followed by the existing quantization, hashing, and sketching steps in RawHash and RawHash2. The index contains hash values from raw nanopore signals instead of reference signals. From matching hash values, we identify many highly accurate chains and report these chains as all versus all overlapping pairs. These overlapping pairs can be directly used by the off-the-shelf assemblers to construct de novo assemblies. We compare the overlaps generated by Rawsamble with those generated by Minmap2 on the forward strand. For base calling, we use various Dorado models on both CPUs and GPUs. We use the Miniasm assembler off the shelf to construct and evaluate de novo assemblies from raw signal overlaps using real datasets with various configurations. We first look at the performance results in terms of runtime for both Rawsamble and the Dorado base caller with different configurations followed by Minimap2. The y-axis shows runtimes normalized to Rawsamble's runtime across different datasets on the x-axis. We find that Rawsamble provides an average speedup of 16 times compared to the configuration when using Dorado's fastest CPU model, and an average speedup of 2 times compared to the configuration when using Dorado's conventional GPU model. Next, we look at de novo assembly construction. This figure shows the visualization of the assembly graph we construct from raw signal overlaps. The biggest takeaway is that we construct the first de novo assemblies ever constructed from raw signal overlaps without base calling. These assemblies are not necessarily short. For example, we can construct contigs of half the E. coli genome length, which is 400 times longer than the average read length for that particular dataset. We conclude that new directions in genome analysis can be enabled without base calling. With this, we cover all our contributions. To conclude, we state that we can mitigate noise in sequencing data and analysis by building a better understanding of the types of noise and developing new algorithms and techniques that can tolerate and reduce noise. We tolerate noise with Blend and understand and reduce noise with RawHash and RawHash2. By doing so, we provide accurate, scalable, and real-time analysis of sequencing data and enable new applications in genome analysis, which we demonstrate by enabling de novo assemblies from raw signal overlaps. We identify 3 key future research directions. First, to improve the base calling performance and accuracy, we should guide base calling with raw signal analysis by filtering reads with raw signal analysis before base calling and using overlapping signal information to train and use base callers for improved accuracy and performance. Second, to improve the accuracy of genome analysis, we should aim to perform full genome analysis without base calling by designing specialized assemblers for raw signals and performing downstream analysis up to variant calling. Third, to improve the performance and accuracy of read mapping, we should rethink heuristic techniques in read mapping as generating highly accurate reads becomes a common practice by using very long seeds with fuzzy seed matching capabilities and enabling reference-free comparisons through accurate de novo assembly constructions. During my PhD, I was fortunate to be involved in many projects from our research group, including 4 my other first and co-first author publications outside the scope of my thesis. We addressed a variety of problems in genome analysis. I would like to thank my advisor, Onur Mutlu, my committee members, and the chair, as well as funding agencies, industry sponsors, colleagues, mentors, collaborators, and friends worldwide. A special thanks goes to my family, my parents, Emine and Turan, and my wife, Çiçek. This concludes my talk.